Welcome back to Botany class. Previous class we concluded with types of plant tissue culture. There are 4 different types: organ culture, meristem culture, protoplast culture and cell suspension culture. So, previous class we concluded with meristem culture. Today let's continue from protoplast culture and cell suspension culture. So, today you are going to learn these 2 questions, very important question. Protoplast culture, 5 more question. Protoplast culture. What's meant by protoplast? Protoplast is the cell without cell wall but bound by plasma membrane. So, if you take a plant cell, this is the plant cell which contains cell wall. Inner to this there is a plasma membrane. So, protoplast is start from this plasma membrane. Inner to plasma membrane there is a nucleus, endoplasmic reticulum, large vacuole in plant cell, mitochondria, here nucleolus, mitochondria, chloroplast. So, all these organelles, cytoplasm. So, protoplast is removal of the cell wall. When you remove the cell wall, what is present inside? Inner to the plasma membrane, along with plasma membrane, inner to the cell what is present is called protoplast. So, from protoplast culture, for protoplast culture, we are isolating this protoplast. There are different steps involved in protoplast culture. So, select this protoplast, a single protoplast, and make it to culture into whole plant. To develop entire plant from this single protoplast is called protoplast culture. There are few steps involved in the culture of protoplast. They are isolation of protoplast. The first step involved in this is isolation. First have to isolate this protoplast from a part of the plant. Then fusion of the protoplast. Allow this protoplast to fuse and culture this protoplast and selection of somatic hybrid. For taking this protoplast, there is a small bit of plant part is taken. Look at this diagram. This is the young plant. From this young plant, any part of the plant can be selected to isolate the protoplast from this organ. Here it is selected as the leaf. Leaf is the small bit of the plant. This is the plant. From this, a single bit of plant part, that is leaf, is selected. Isolate the protoplast from the leaf part. So, to take out this protoplast from this leaf is immersed with the enzyme mixture. So, what are the enzymes used for immerse this leaf? Here this is the sterilization, leaf sterilization. The leaf is sterilized with enzyme mixtures to isolate the protoplast. So, first step is isolation of protoplast. So, this selected leaf is immersed with 0.5% macrozyme. The enzymes are important. 2 mark and 3 mark question it will be asked. 0.5% of macrozyme, 2% of Onotsuka cellulase enzyme, cellulase, and 13% of either sorbitol or mannitol, and the pH maintained at 5.4. So, this enzyme mixtures, the leaf can be immersed, and overnight this can be incubated with 25°C to soften this leaf. And after, now this leaf become soft, and from this leaf the epidermis of this leaf can be peeled off. When you peeled off this leaf epidermis, you may get this type of peeled leaf segments. here the cells can be visible, and from here this leaf segment peeled, from this peeled leaf segment the protoplast can be isolate. A single protoplast can be isolate. So this is the plasmolized cell. The protoplast comes out. When you peeled off this epidermis, the protoplast comes out, and now this process is called as isolation. So first step is isolation of protoplast. After isolation, the cells again it immersed with some enzyme mixtures. So it can be put it into different enzyme mixtures to remove the cell wall. So now the cell is isolated with cell wall. So make this for centrifugation. When you make it to centrifuge, the cell wall digestion, where this cell wall has to digest. So now this cell wall can be removed and isolate the protoplast from this. So to removing this cell wall from this, it's again this cell wall digestion by centrifugation. So when you allow it to centrifuge, it can be washing out the debris and the protoplast. The cell wall comes as a debris and the pure protoplast can be isolated. So from here to there, the steps involved in this is isolation. So after centrifugation, the pure protoplast can be isolated. Now second process is fusion of protoplast. This protoplast can allow to fuse. So this protoplast, it allow it to fuse. So fusion of, here it produces a somatic hybrid. Here the hybrid produced is named as somatic hybrid. How to obtain this somatic hybrid? So next process is fusion. For making fuse of this protoplast, it's treated with fusogenic agent. Normally the fusogenic agent is PEG, polyethylene glycol is the normal fusogenic agent. So allow you to this PEG. When you treat it with this 25 to 30% of polyethylene glycol along with Ca++ ions, this fusion takes place. So it just joins this protoplast, it just joins. Here the cell, here the cell wall is absent. Now everything is removed by centrifugation. Now this plasma membrane which joins together, and now this fusion allow it to fuse by plating technique or this allow you to micro drop array. So micro drop array or this plating technique, the protoplast are allowed to fuse. So fusogenic agent is first fusogenic agent allow you to fuse. After that, when it is culturing, it allow this fused protoplast are allowed to culture to produce a clump of tissues. So these techniques are used, plating technique or this micro drop array technique is used for making regeneration of the cell wall. So this process is culturing. So third step is culture of protoplast. First isolation, then allow it to fusion with PEG. Normally we call this as PEG, polyethylene glycol, and culture of protoplast. To culture it, this technique can be used, micro drop array or this plating technique. So this is transferred to MS medium. So culturing this MS medium, Murashige and Skoog medium, MS medium with little modifications in the droplets and allow this plating technique or this micro drop array technique. And now these are allowed to produce a clump of cells. So from this few cells, the cell wall already removed and regeneration of cell wall. Now after culturing, when it is cultured, the cell wall has to regenerate. Here it starts to regenerate and it produces a clumps of cells. So from this clump of cell, there is a callus formation. So what is callus? Callus is a mass of unorganized tissue. So mass of unorganized tissue is developed. From this callus, differentiation starts. So callus differentiated and differentiation of callus. So it produces organogenesis, followed by organogenesis. The shoot, root, everything, it reappears, and finally it produces the plantlet. So when it produces this, the final step is selection of somatic hybrid. Here the hybrid, after fusion, the hybrid will be formed. This hybrid is called as somatic hybrid. So at first it produces as a cybrid. What is cybrid? Here two more question is there. Cybrid and somatic hybridization. Cybrid is formed when this cell fused. The plasma membrane, just randomly it fused together. Now this is without involving nuclear fusion, it's called cybrid, followed by nuclear fusion. Then this nucleus fused together. Now this process is called as somatic hybridization. So from the protoplast, without nucleus, the fusion, it produces, the byproduct is cybrid, and this is followed by this fusion called as somatic hybridization. So now it produces a somatic embryo or embryoid will be developed, and from this organogenesis, this plantlet is developed. So it develops as a young plant. So this plantlet derived from protoplast and it grows as a young plant after hardening. So this is the process of protoplast fusion. So first question is protoplast culture. Culture of protoplast. This is one of the type plant tissue culture. Another question, another type of plant tissue culture is cell suspension culture. What's this? Cell suspension culture. So cell suspension is nothing but a single cell or a clump of cells. It's transferred to a liquid medium and make it to produce as the entire plant. So this is developed from the callus tissue. You know that callus is a unorganized mass of cell. So mass of cells are present there. So this mass of cells can be separated. Separate by shakers. Allow you to shakers to separate this callus tissue. From the callus, few cells or single cell can be selected and it is make it to culture in a liquid medium. That culture is called cell suspension culture. So there is a difference between protoplast culture and cell suspension culture. What's the difference? In protoplast culture, from the cell, after removing the cell wall, this protoplast is isolated and allow you to culture. This method. Cell suspension culture, from the callus, a few cells or clump of cells can be selected with shakers. From the shakers to allow it to separate, and it is transferred into a liquid medium to make as a culture called cell suspension culture. So this cell suspension culture is used for the production of secondary metabolites. What are secondary metabolites? The plants which produces chemical compounds or chemical substances which is not used for the normal growth and development of the plant. So it gives as a byproduct is called as secondary metabolites. There are number of secondary metabolites can be used. There are terpenoids, alkaloids. So this all comes under secondary metabolites. So these secondary metabolites are taken from the plants as a byproduct. So commercially it can be culture it in the bioreactor. Commercial production, this secondary metabolites, because it's used for pharmaceuticals. Economically it's important commercially. So large scale preparation by this bioreactor. So, a few examples are given below. What are the examples of cell suspension culture? Here there is a product called digoxin. This is the product, that is secondary metabolite. This is the source of secondary metabolite from which plant it obtained, and this is the uses. This also important. There are 5 examples are there. Sometimes it may ask as a 5-mark question. So learn, this is the product or secondary metabolite: digoxin, codeine, capsaicin, vincristine, quinine. So number 1, digoxin, codeine, capsaicin, vincristine, quinine. So this digoxin is obtained from Digitalis purpurea. Now whenever you write this binomials, underline separately the genus name and the species name. The digoxin is obtained from Digitalis purpurea. What's the use of this? It's used to give cardiac tonic. So to treat heart disorders, this is used as a cardiac tonic. Digoxin. The product or secondary metabolite is digoxin. Digitalis purpurea. The source, the plant source is Digitalis purpurea. Use is cardiac tonic. It's used as a cardiac tonic. Another one, codeine. The source of codeine is Papaver somniferum. Papaver somniferum. This use is analgesic. It's used as an analgesic. Then capsaicin obtained from Capsicum annuum. Capsicum annuum. The use is rheumatic treatment. It is used to treat rheumatic pain, rheumatic treatment. Then vincristine. Vincristine is obtained from Catharanthus roseus. Catharanthus roseus, you know, common plant. So this Catharanthus roseus, it's an anti-cancerous one substance, anti-cancer, carcinogenic, anti-carcinogenic. So to treat cancer, this vincristine is used. And quinine. Quinine is obtained from Cinchona officinalis. It's used to treat, it's treat as anti-malarial substance. It's used for anti-malarial. So first name the product. There are digoxin, codeine, capsaicin, vincristine, quinine. So from the name itself, you just remember this name, digoxin, which is obtained from Digitalis. Digoxin digitalis. Digitalis. Remember, digoxin digitalis. Codeine, Papaver somniferum. Then capsaicin, digoxin digitalis. Capsaicin, capsicum. Capsicum annuum. And vincristine, Catharanthus roseus. Quinine, Cinchona officinalis. Cinchona officinalis. The quinine is obtained from Cinchona officinalis. What are the uses? Digoxin used to treat cardiac. It's used as a cardiac tonic. Codeine used as analgesic. Capsaicin used to treatment of rheumatic. It is used for rheumatic treatment. Vincristine, it is anti-carcinogenic. Quinine, anti-malarial. So this is given in a tabular column in your book. So second question you are going to learn today is this question, cell suspension culture. This is only the example. First you learn what is cell suspension culture. Here protoplast culture. Here 4 steps involved in protoplast culture. Next question is plant regeneration pathway. Plant regeneration pathway. So plant regeneration pathway, here explant is important. What's the explant? A part of the plant, isolated part. Here the explant is leaf. So explant can be selected any part of the plant like meristem or the shoot apex or stem, leaf, root, or callus, any part of the plant is used as a explant. Here meristem and shoot apex, these are used to produce virus free plantlet. So virus free plants are, if any viruses are invade through the root, we can select the meristem region, the shoot apex of the, so this region, shoot apices can be selected and culture it. So this virus cannot enter into the plant. Likewise, virus free plantlet can be produced in this plant regeneration pathway. And this stem part, if you select as a stem part, the nodal region, from the nodal region it is used to micropropagation, that is multiple shoot formation produced by this stem or nodal part. So from this multiple shoot, it makes to index shoot induction. So shoot induction or root induction, and from the root induction, root induction is induced to produce the root, and induced to produce from this root and shoot formation, it produces a plantlet, and the plantlet is, next process is hardening. So after this plantlet develop, it is till the plantlet development, it is in the lab or within the glass vessel. Later it can be hardening is the process, stepwise process to expose it to the direct environment, that is hardening. Or this explant from the callus formation, callus, if it produces a callus, from the callus there is indirect embryogenesis, or from this explant, direct embryogenesis, that forms as a embryoid, somatic embryoid, and plantlet develops, and the plantlet is then next step is hardening. So this plant regeneration pathway is classified into two different types, that is somatic embryogenesis and organogenesis. So this is somatic embryogenesis, somatic hybridization. It produces embryoid. Somatic embryo is developed, it is called as embryoid, because from the callus directly it produces a somatic embryo, that is called embryoid. From callus embryoid is developed, is called as somatic hybridization. Another one, organogenesis. In organogenesis, how it produces? Organogenesis. Here, organ. At first, this callus is developed. Callus. From this callus formation, so this is organogenesis. From the callus, the differentiation. Here, differentiation. It may be differentiated into either shoot or root. So organogenesis is the formation of this callus. From the callus, it differentiated into either shoot or root. If it produces the shoot, the process is caulogenesis. Or when it produces the root, it is known as rhizogenesis. So caulogenesis and rhizogenesis, both of this caulogenesis and rhizogenesis, it produces a plantlet. So this plantlet can be transferred to hardening, so direct environment. So this process is organogenesis. First, somatic embryo, so regeneration pathway, plant regeneration pathway classified into two, somatic embryogenesis and organogenesis. Somatic embryogenesis, from callus embryoids are developed. Here, organogenesis, from callus organs, differentiation taking place. So parts are differentiated, either shoot or root. If shoot develops or derived from the callus is called caulogenesis. Root derived from the callus is known as rhizogenesis. And this shoot and root, it produces a plantlet. Then the last question, what are the applications? What's the applications of this protoplast culture? It's used to produce new variety. So, improved hybrid production, improved hybrid varieties, that is disease resistant, virus resistant varieties can be produced by this protoplast culture. So, here virus free resistant, here when it is regenerated, when it allowed to regenerate, the virus free plants to produce it, make use of the shoot apices, apex cells, apical meristem culture, virus free, that is one of the application. Then somatic embryo can be developed, or syn seeds or artificial seeds can be produced by this. This is used for conservation of biodiversity, to conserve the biodiversity. There are so many rare plants which cannot grow in the natural environment condition or normally. So, this technique can be used for the conserving the plant diversity in this protoplast culture. Then so many stress resistant varieties are obtained. Stress resistant varieties, virus resistant variety, stress resistant variety, and it is also useful in forestry. The micropropagation, so many plants are developed to maintain forest, it is used. So, application of this is very important. And finally, secondary metabolites can be produced. Secondary metabolites. Secondary metabolites, you learn some examples here, digoxin, codeine. Not only this is few examples, so many secondary metabolites from the chemical compounds of this plants, it used as a byproduct, taken as a byproduct. Commercially, it's very important. So, what's the last question is applications of protoplast culture. What are the applications? Disease resistant varieties or virus resistant varieties, stress resistant varieties can be produced, and the somatic embryos or artificial seeds. So, these seeds are encapsulated in the embryos are encapsulated, enclosed seeds. So, these seeds are called as syn seeds or artificial seeds, can be produced by this technique, and this stress resistant secondary metabolites, and it is also used to conserve the biodiversity, and in the forest micropropagation is very much useful in forest. So, first question, let us review protoplast culture. Today's question you learn, first question protoplast culture, second question cell suspension culture, third question plant regeneration pathway, last question applications of protoplast culture. Learn all this question well. Thank you.